Good morning. Good morning, everyone. 11:07 a.m. in the morning, Monday, October 30th, year 2023. Only one more day left in the month, being tomorrow, being Halloween. Just adjusting a few sound levels here, trying to get rid of a little bit of feedback. And today we're going to finish up. We have to follow it through. Finish up this reading of the Milstein, Cesar Milstein antibody article. Several distractions have come up recently, but the proper thing to do is to follow it through, see it to its completion. I noticed that our friend John O'Loughlin is a little bit tied up today, needs a day off. So I figure for those of you who follow my show, several of you are multiple of you, that's the right way to say it, are also fans of O'Loughlin. So to fill in your time, I had a little bit of an opportunity here. It's raining outside, and to finish up this reading. So without any further ado, this is 207. No. What would I give this episode to? I think it's 245. C. Yeah, and we had 3 different little addendum episodes mixed into here as well. Good morning, RT3. Good to see you. All right, so without any further ado, let's just get into it. So this is the second to the last chapter. It's titled Milstein's Legacy. Milstein's Legacy. First subsection is technology for healthcare. So in 1980, Cesar Milstein wrote, quote, "All that we seem to have acquired is the potential ability to select from an animal any of the antibodies of his repertoire. It is something like selecting individual dishes out of a very elaborate menu. Antibodies à la carte. A gastronome worth his salt wants to experiment with new ingredients, new combinations. His dream is to invent new dishes and not only to taste what others are doing. I am sure our next step will be to move from the dining table where we order and consume our antibodies à la carte to the kitchen where we will attempt to mess them up," unquote. He was quoted saying that in the 1980 Wellcome Foundation lecture. As these words suggest, from the time that Milstein first developed the hybridoma technique with Kohler, or Georges Kohler, he began looking for ways to improve the technology. Let me step out of the way a little bit. Hello, Marie. Hello, Rona. Hello, RT3. Big picture here. You can see this is how they describe their manufacturing of these monoclonal antibodies, these selected antibodies. By the late 1970s, most monoclonal antibodies were being generated by using the protocol in the diagram, which we just showed above. Such monoclonal antibodies are produced by fusing animal myeloma tumor cells with spleen cells, as we read earlier, derived from mice or rats previously immunized with an antigen. Monoclonal antibodies created this way are known as murine monoclonal antibodies, denoting that they have their rodent origin. The advantage of murine monoclonal antibodies was that they could be made to target almost any antigen and in vast quantities. Indeed, the rodent system possessed great flexibility to generate an antibody with any number of determined specificities. The system, however, has a downside. It produced non-human antibody molecules. Seen as foreign by the human body, such antibodies ran a risk of promoting adverse immune reactions if administered to patients. Moreover, these adverse reactions could result in the rapid destruction and clearing of the monoclonal antibody itself from the body, which reduced their effectiveness for therapy. Early on, Milstein and his colleagues had attempted to generate human monoclonal antibodies. This, however, had proven both very difficult and impractical. Manifestly, humans could not be immunized and manipulated like laboratory animals. Sorry. immunizing humans with cancer antigens for the purpose of raising human antibodies against cancer to be converted into monoclonal antibodies, for example, was full of risks and raised serious ethical concerns. Not to mention the fact that a cancer is what they're trying to target a cancer cell. Sometimes cancer cells can't be identified by their antigens, just by some outside molecule as a foreign exosome or a virus, if you will, or whatever. Making monoclonal antibodies a la carte. By the late 1970s, when scientists were grappling to produce more human-like monoclonal antibodies, Cesar Milstein began to encourage his colleagues to think about using recombinant DNA, rDNA, or genetic engineering to solve the problem. That is very, very interesting. First devised by Stanley Cohen at Stanford University and Herbert Boyer at the University of California in 1973-1974, the technique involved using restriction enzymes to isolate a DNA fragment containing a gene of interest from an animal cell and then fusing this with a bacterial plasmid, an independent self-replicating DNA molecule that had been cleaved open with restriction enzymes. This method created what was called a recombinant DNA molecule, or rDNA. Once made, the molecule could be put into a bacterial cell for amplification alongside the host cell DNA. Milstein argued that by combining genetic engineering with hybridoma technology, scientists could shift from being restricted to immortalizing naturally occurring antibodies, as he and Kohler had done, and begin to design tailor-made antibodies. He, Cesar Milstein, believed this method could provide a way of engineering the human antibodies scientists had so far failed to produce. One of the advantages scientists had in using genetic engineering to improve monoclonal antibodies was that antibodies possess a basic, uniform, modular structure. This meant that scientists could cut genes present on one part of the antibody and paste them on the constant region of another antibody. For many scientists steeped in antibody research and immunology, such a shuffling of genes merely mimicked what had already happened naturally in the immune system. Cesar Milstein himself would make relatively modest efforts in the application of genetic engineering to monoclonal antibodies. Nonetheless, his vision inspired many others to begin the process. This included Michael Neuberger, Neuberger, and Gregory Winter, based at the Laboratory of Molecular Biology, also in England. They were instrumental in developing techniques for re-engineering monoclonal antibodies. This helped reduce their non-human protein components, thereby enhancing the efficacy and safety of monoclonal antibodies for use in therapeutics. With Milstein's encouragement, the process of making monoclonal antibodies underwent a radical transformation over the next few years, yielding antibodies that were much more compatible with the human body. By the 1990s, scientists had gone a long way towards remolding the basic monoclonal antibody first developed by Cesar Milster and Georges Kohler and Cesar Milstein. And who's in this photo here? We got Milstein at the far left, Michael Neuberger in the middle there, and this guy on the far right is that Gregory Winter. Just hanging out casually at a bookstore, I guess, that sells nothing but books on genomics. With antibody engineering, scientists and clinicians found a way of producing a much safer and effective form of monoclonal antibody, one that could be used as therapeutically for many different diseases. Today, monoclonal antibody drugs account for a third of all new treatments being introduced worldwide. Again, this is an article from 2012, and are beginning to bring in far greater revenue per product than older and more traditional small molecule pharmaceutical products. They have also been more successful in completing clinical trials than small molecule drugs. A form of a small molecule drug, by the way, would be remdesivir. Interesting. So, and another antibody, although maybe it's not monoclonal technically, or I'm not sure, is ZMapp. Learning from past mistakes, both the Laboratory of Molecular Biology and Medical Research Council ensured patents were filed for the re-engineered forms of antibodies generated. This has generated an important source of revenue. To date, the estimated MRC, again, that's the Medical Research Council, net income from monoclonals as of 2012 was $360 million after deduction of revenue share and awards to inventors. In 1975, Milstein was elected a Fellow of the Royal Society. 2 years later, he was awarded an honorary doctorate by the Universidad Nacional del Sur, based in the city of his birth in Bahia Blanca, Argentina. This was given in recognition of his contribution to science. Cesar Milstein received several other awards after this, including the Wolf Prize in Medicine in 1980, the Royal Medal in 1982, the Lasker Award in 1984, and the Copley Medal in 1989. He was jointly awarded the Nobel Prize in Physiology or Medicine in 1984 with Georges Kohler and Niels K. Jerne, Jerne, who, by the way, also did not live much past, I think he died in 1990 or 91, only in his 50s as well. Jerne, quote, for theories concerning the specificity in the development and control of the immune system and the discovery of the principle for production of monoclonal antibodies was the name of the joint, how would you say? That was what these three men were awarded the Nobel Prize for. By the time the Nobel Prize was awarded to Milstein and his colleagues, monoclonal antibodies had become a ubiquitous method for researchers in a multitude of disciplines. The technology not only provided the tool for those involved in basic science, but also for those looking to develop new diagnostics and therapies. Some idea of how valuable the technique had become can be seen by the fact that as early as 1980, the British biotechnology company called Celltech, Celltech, calculated the world market for monoclonal antibodies would soon reach $6 million. 4 years later, in 1984, the United States Office of Technology Assessment indicated that the United States market for monoclonals was worth between $5 and $6 million, predicting it would rise to between $300 and $500 million, half a billion, by 1990. Yet the rise of monoclonal antibodies had not been a straightforward or inevitable process. It had taken years of work on the part of scientists to demonstrate its utility. Milstein himself had been at the forefront of the process. Realizing he could not leave such a task to others, Milstein quickly shifted temporarily from his longtime basic scientific interest in understanding antibody diversity to exploring, with many collaborators, the practical applications of monoclonal antibodies. We see here, I still have a stamp collection, pretty modest by most most collectors. Argentinian stamp in 2005 celebrating Milstein. Conscious of his own difficulties as a scientist in Argentina, throughout his career, Milstein devoted himself to assisting science and scientists in less developed countries. In March 2000, he wrote trenchantly, quote, "Science will only fulfill its promises when the benefits are equally shared by the really poor of the world," unquote. Science will only fulfill its promises when the benefits are equally shared by the really, really poor people of the world. Well, I don't know. I don't know how you guys feel about that. Seeing that it looks as though sometimes the most extreme poor people of the world are used for research. I don't know. In recognition of his legacy, the MRC later created a doctoral studentship in Milstein's name after his death to help support Argentinian scientists who wish to pursue their studies in Cambridge. Milstein died in 2002 from a heart attack, age 74. First diagnosed with cardiovascular problems in the 1970s, he adhered thereafter to a strictly controlled diet and pursued regular exercise regime. Colleagues recall seeing him taking his regular walk around Cambridge. Accompanied by his dog, Milstein would either take a portable radio to listen to the news or dictate letters onto a tape recorder while walking. Often he would invite colleagues to accompany him on his walks to discuss their work. And the last section is glossary. So you can view the glossary of terms from there. And that pretty much wraps it up. Very important person historically, Milstein. There was a research paper done by the star of the show last night, Jay Berzofsky. I wonder if I could actually find that. So this was a presidential address. Presidential address to the American Society of Clinical Investigation in April of 1994, given by that Jay Berzofsky regarding cross-fertilization among fields, a seminal event in the progress of biomedical research. So he talks about the different major inventions, progress, breakthroughs in biomedical research up until that time in 1994. And I found it very interesting. And the only reason I found this article was I was trying to find some stuff on Berzofsky's father, Max Berzofsky. And in this image, you can actually see almost a core picture right here, core to many of these research, or I should say scientific breakthroughs in biomedical research, is the Milstein-Kohler antibody, monoclonal antibody research right there, of which a whole bunch of other things fed into or were derived from, for immunology Nobel Prizes, partial genealogy. So it was recognized that this was a seminal piece of work by someone who was, again, a star of our show last night for our weekend show, Jay Berzofsky. I'll try to leave that on that show on YouTube for a little while longer. But it's a little spicy, so I'll have to take it down soon. Let's do a quick check-in with you guys. Let's see here. Can I add in... Why is that not working? I think I'm going to have to close and restart that. Okay, very good. Now I can see the chat. Hello, X-Ray Girl, RT3, Laura, Marie, Mobster. We have overcome the ethics issue. It was all in our head. Yeah, and now the issue is preventing this rewriting the history of what just happened. And you can see that there is people are being tricked into a position of, you know what? Thank goodness COVID happened, because it allowed us to roll out these amazing new therapies, which will work with a little bit more tuning and a little bit more control at the factory. That's the new assessment that we are all being encouraged to take. Not let's revisit the conditions by which this stuff was approved in the first place, but instead let's focus on how it could have been made a little bit better. Let's focus on that. Let's ignore what led us there. And the continuous attempts to not even talk about that, not talk about the underlying conditions, just keep going straight to, well, we found out why it's a problem. Well, you know what? If none of it was needed in the first place, then all of it's a problem, period. But anyways, that's getting off the topic here. But anyways, this was a quick video. I just wanted to wrap this up. I wanted to read that last chapter. We'll finish this. We'll put this on the playlist. I may do another show later on. And just to give you guys a few more updates, maybe we'll just do open Q&A. I don't know. But that said, thank you very much again, everyone, for your support. It was great to be able to read this series. I will probably listen to it again myself, because sometimes when I'm reading, I'm not even quite processing all of it, of what's going on. And I think what we've learned yesterday, especially regarding the life's work of Berzofsky and the 20, 30, 40, 50 years of research for vaccines for cancer, is that when you're targeting whole cells and you're targeting cancer, I never really appreciated this until yesterday. It's a whole new ball game. It's a whole new ball game. And yes, they want to use the avenue of vaccines for cancer treatment. Vaccines are targeted by this, defined by this whole world of antibodies, which may not really map that well at all to cancer treatments. But they've been trying to make it. They've been trying to force one shape into another shape. So with that said, interesting stuff. We'll do more on that topic soon enough. I appreciate all you guys for everything you do, all your support. Take care. God bless. And we will be in touch again soon.